Hello and welcome to MitoTalk. Let's have a deep dive in Complex 2 and the TCA bridge. Hello, welcome back. Your destination to all things mitochondria. I'm your host, Dr. Sukant Khurana, and today we continue with our deep dive series on electron transport chain. In our last episode, we explored Complex 1, the gatekeeper of electron flow. Today, we are moving to Complex 2, also known as Succinate Dehydrogenase, a fascinating molecule that connects two essential cellular processes, the TCA cycle and the electron transport chain. Thus, again, is our guest expert, a budding young biologist, Basudha Banerjee. Welcome back, Basudha.
Thank you very much, Dr. Sukant. I am happy to return and excited to unpack this complex to the multitasker of the mitochondria.
Well, let us start with basics. Complex 2 is often overshadowed by Complex 1, but it has a unique identity. Could you break that down for us?
Absolutely. So, complex 2 or succinate dehydrogenase is the only complex in ETC that is also a part of the TCA cycle. It catalyzes the oxidation of succinate to fumarate, generating FADH2, which in turn donates electrons to the ETC via ubiquinol. And unlike complex 1, it does not pump protons across the mitochondrial membrane. So it contributes to the electron flow, but not to the proton gradient directly. Think of it as a bridge, like relaying signals between energy metabolism and respiration.
Well, let's look at subunit structure and function. What about its architecture? I hear a lot about subunit mutations. How is it organized?
Basically, complex 2 has four core subunits. SDHA, where succinate is oxidized, SDHB, which contains the iron sulfur clusters primarily for the electron transfer, and then we have SDHC and SDHD, which help to anchor the complex to the inner membrane and pass the electrons to the ubiquinol. And mutations in any of these, especially the SDHB, which is associated with inherited tumors like paragangliomas and pheochromocytomas. What's unique is that these tumors are metabolically driven, and that is what makes complex 2 so medically interesting.
Well, let's talk about the disease link, complex 2 in diseases. It seems to have a very different profile compared to complex 1.
That's absolutely right. So, while complex 1 dysfunction is linked to energy failure and ROS, complex 2 issues often create metabolic signaling disruption. And loss of complex 2 leads to the accumulation of succinate, which acts as an oncometabolite, stabilizing HIF1 alpha, which is a transcription factor that generally promotes tumor growth under hypoxic conditions. We see this in several hereditary cancers. On the other side, neurodegenerative conditions like the Leigh syndrome can also involve complex 2 through a it is less commonly involved than complex 1. It is a versatile actor causing both oxidative stress and pseudohypoxia.
Well, let's look at therapeutic challenges as well as the targets. This all that you mentioned, think of it as opens a lot of doors for therapeutics. Are there viable drug targets within complex 2?
There are, but they're very tricky because complex 2, since it does not pump any protons, manipulating its effect, metabolic signaling more than energy directly. One exciting direction is targeting the succinate accumulation and its downstream effects. There's work on succinate receptor blockers like the SUCNR1 antagonist and molecules that modulate the subunits iron sulfur clusters. And another interesting idea is reversible inhibitors that modulate complex 2 in cancer without harming the normal cells. It's delicate because you don't want to crash the energy supply.
Well, this is very interesting. Now let's look at AI tools sort of for mutation mapping. Let's switch gears for complex 2. Is AI being used to predict pathogenic mutations in this complex?
Absolutely. And it's incredibly promising. We use deep learning algorithms trained on structural and clinical mutation databases, which help us to predict which SDH mutations are oncogenic and others that are benign. AI is also helping us to simulate any conformational changes in the binding sites and the iron sulfur cluster geometry, which is normally very hard to study experimentally. And in the future, we might see personalized interventions based on predicted SDH mutational landscapes from cancer to mitochondrial disorders.
Okay, so let's look at the bigger picture. Before we wrap, I'll always like to zoom out. Why should someone care about complex 2?
Because it is more than just a cog in the ETC. See, complex 2 is where metabolism and respiration shake hands. It teaches us that mitochondria aren't just the energy machines, they are sensors and regulators. They tell the nucleus how the cell is doing. Understanding complex 2 can help us treat cancer, neurodegeneration, and a lot of metabolic disease with just one target.
Well, beautifully said, as always, Basudha. Thank you for this insightful session. To our listeners, keep feeding your curiosity and your mitochondria. Next in our deep dive, Complex 3, the middleman with a double life. Until then, this is MitoTalk. I'm Dr. Sukant Khurana. With me was brilliant Basudha Banerjee. Stay energized and informed. Thank you.
Thank you so much.